Now, in today's discussion, we're going to consider the citric acid cycle. The citric acid cycle is part of the oxidative um, mechanism for the extraction of energy in the form of ATP from glucose and from other fuel sources. The citric acid cycle is intimately associated with the electron transfer chain. All right. For now, we're going to focus our attention on the citric acid cycle, the reactions involved in it, its regulation, and its role in metabolism. Alright, previously we have considered glycolysis and other carbohydrate metabolic pathways. We've learned that in glycolysis the yield of ATP is just 2 molecules of ATP per glucose molecule, and we attributed this relatively small gain or yield in ATP to the fact that the reaction, the oxidation of glucose to lactate in the anaerobic glycolysis, um, to the fact that this process is incomplete in the sense that the molecule of glucose is incompletely oxidized into into um, into molecule of pyruvate and ultimately into lactate. You're going to notice the glucose is a 6 carbon sugar, and in the oxidation to pyruvate or lactate, you ended up with just 2 3 carbon sugars, and therefore the molecule indeed is not completely oxidized. A very small and only a very small fraction of the energy, the chemical energy contained in the molecule of glucose, has been extracted and released from the molecule. In the citric acid cycle, the molecule of glucose is completely oxidized to carbon dioxide. Remember, carbon dioxide is the most oxidized form of carbon. Therefore, the conversion of glucose to carbon dioxide entails the complete oxidation of glucose in this case. Okay, the citric acid cycle has a central role in metabolism. In fact, if you're going to map out the entire metabolic pathways, you're going to find the citric acid cycle will be somewhat at the center. Right? The citric acid cycle is a catabolic pathway, but it's also an anabolic pathway. Three processes play central roles in aerobic metabolism. First is the citric acid cycle. Second, the electron transfer chain, and then the oxidative phosphorylation. We're going to discuss electron transfer chain and oxidative phosphorylation in the next session. For now, we're going to consider the citric acid cycle. Now, the citric acid cycle is also known as the Krebs cycle or the tricarboxylic acid cycle. Krebs cycle in honor of Hans Krebs, who has discovered this pathway, um, the tricarboxylic acid cycle, because the fact that the citric acid cycle consists of um, some some compounds with 3 carboxylic acid groups as intermediates. The metabolism, remember, consists of catabolism, it's the oxidized breakdown of nutrients, and anabolism, just a reductive synthesis of biomolecules. As I mentioned a while ago, the citric acid cycle is both a catabolic and an anabolic pathway. In other words, it's amphibolic because it plays a role in both catabolism and anabolism, and it is the central metabolic pathway. Alright, now this is a diagram of how exactly we get ATP from pyruvate in the process of oxidative respiration. Oxidative respiration again consists of the Krebs cycle, the electron transfer chain, as well as oxidative phosphorylation. Now let me use my pointer. Alright, so first is that remember that the pyruvate is formed from the oxidation of glucose in anaerobic glycolysis, or should not say anaerobic glycolysis, I should say in glycolysis. Anaerobic glycolysis, remember, is when glucose is converted into lactate. So in glycolysis, pyruvate is formed from glucose. The glucose, um, fatty acids, even and amino acids are oxidized into um, acetyl CoA, like glucose is oxidized to pyruvate, which is then oxidized with a pyruvate dehydrogenase complex that undergoes decarboxylation, producing the first carbon dioxide out of pyruvate, the process converting it into acetyl CoA. Fatty acids also oxidize into acetyl CoA. Amino acids, their carbon framework are oxidized into acetyl CoA. These acetyl CoA molecules um, enter citric acid cycle. So citrate, then citrate, when it's the intermediate, it's not citrate itself, but the intermediate undergoes oxidative decarboxylation. So here goes our second carbon dioxide molecule out of pyruvate. The remaining third carbon dioxide accounts for the third carbon atom in pyruvate. Um, Here you lost it here, and the steps from the production of acetyl CoA to some of the steps in the citric acid cycle produce reducing equivalents, typically nicotinamide adenine dinucleotide (NADH) or flavine adenine dinucleotide (FADH2), and this NADH and FADH2 are oxidized in the electron transfer chain in oxidative phosphorylation, coupled to oxidative phosphorylation in the process, yielding adenosine triphosphate or ADP and inorganic phosphate. What you ought to bear in mind is that the ultimate electron acceptor in this case is oxygen. That's why it's called aerobic respiration. Alright, so here are the three important steps in the process of oxidative respiration. Again, the oxidation of pyruvate to acetyl CoA, the oxidation of acetyl CoA in the citric acid cycle, and then the electron transport as well as oxidative phosphorylation. Alright, now where does the citric acid cycle take place? It takes place inside the mitochondrion. Alright, process of glycolysis and the process of gluconeogenesis. Um, Except for some steps in gluconeogenesis, specifically the conversion of pyruvate into phosphoenolpyruvate that occurs in the mitochondria. Glycogenolysis and glycogenesis, and the pentose phosphate pathway, this occur in the cytosol, but um, the citric acid cycle occurs in the mitochondria. So it is important for us to um, remember, or excuse me, consider the structure of the mitochondrion. The mitochondrion consists of an inner and an outer membrane, right? Inner and outer membrane. The reactions in the citric acid cycle occur inside the or within the inner membrane, and that region in space is called the mitochondrial matrix. There is one reaction though um, of citric acid cycle that does not occur in the matrix of the mitochondria, but occurs on the um, uh, occurs on the um, uh, inner mitochondrial membrane itself, rather, because the enzyme that is involved in that reaction is actually an integral membrane protein. Okay. Now, molecules can more or less freely penetrate, go in, and go out of the outer mitochondrial membrane, but only certain molecules are able to cross the inner mitochondrial membrane. Those that have specific protein transporters um, that are able to um, let them in or let them out of the mitochondrial of the inner mitochondrial membrane. The space between the inner and the outer membrane is called um, the intermembrane space, and the intermembrane space will it's important in oxidative phosphorylation will be more apparent in our next discussion. Okay, so these are the features, important features of the citric acid cycle. Again, the pyruvate from glycolysis is oxidized into acetyl-CoA, leading to the formation of carbon dioxide and NADH. Acetyl-CoA reacted oxaloacetate in the process catalyzed by citrate synthase, in the process producing citrate. Now, citrate is then um, isomerized by aconitase into isocitrate. Isocitrate undergoes um, oxidation into alpha-ketoglutarate, process forming second carbon dioxide from pyruvate. The first is obtained from the oxidation of pyruvate to acetyl-CoA catalyzed by pyruvate dehydrogenase. Now, the alpha-ketoglutarate that is formed then undergoes another round of oxidation, producing that second carbon dioxide, and this one NADH again, that's already one, two, three, the third NADH, and then this to produce succinyl-CoA. Succinyl-CoA then um, is converted into succinate by succinyl-CoA synthetase, um, producing GTP. But GTP is just converted into ATP with enzyme nucleoside diphosphate kinase. Right? The succinate is then oxidized further, succinate dehydrogenase to fumarate. Now, the process producing not not um uh, not NADH but FADH2 as reducing equivalent. This is the enzyme that occurs in the inner mitochondrial membrane because succinate dehydrogenase is an integral membrane protein. The fumarate is hydrated into malate by fumarase, and then malate is oxidized by malate dehydrogenase to oxaloacetate, the process completing one cycle, and you form then another molecule of NADH. Right? We're going to consider the details of this reaction now. Okay, the pyruvate dehydrogenase complex is the enzyme that is responsible for the conversion of pyruvate to carbon dioxide and the acetyl portion of acetyl-CoA. Now, it's called a complex because there are actually 5 enzymes that are found in this complex. These 5 enzymes are um, associated with each other, right, to produce a complex system. Now, the 5 enzymes in the complex consist of pyruvate dehydrogenase, dihydrolipoyl transacetylase, dihydrolipoyl dehydrogenase, pyruvate dehydrogenase kinase, and pyruvate dehydrogenase phosphatase. Now, the last two, the kinase and the phosphatase, are enzymes that are involved in the regulation of the activity of the pyruvate dehydrogenase complex. The first one involves pyruvate dehydrogenase is needed in the oxidation of acetyl-CoA. The second is needed for its reaction with um, acet with CoA or coenzyme A, also given the symbol CoAsh. And then lastly, the dihydrolipoyl dehydrogenase is needed for the oxidation of the lipoic acid back into its oxidized form. Okay, first, hopefully, alright, there's this diagram. The first step, the pyruvate loses carbon dioxide and hydroxyethyl TPP is formed. In the second step, so what is catalyzed by the enzyme pyruvate dehydrogenase, right? In the second step, the active form of lipoic acid is bound to the enzyme dihydrolipoyl transacetylase by an amide bound to the epsilon amino group of a lysine. Okay, then dihydroxyethyl group is oxidized and transferred to a sulfur atom of a reduced form of lipoamide, and lipoamide is reduced to dihydrolipoamide. In step 3, the acetyl group is transferred to the sulfhydryl group of coenzyme A. Next, dihydrolipoamide is oxidized to lipoamide. Now, do not worry, I'm going to discuss them in detail now. First step is the loss of carbon dioxide and the formation of hydroxyethyl TPP. So as I mentioned, this is pyruvate. First molecule of carbon dioxide is removed with the process of oxidative decarboxylation. Decarboxylation naturally because carbon dioxide is removed from the molecule, process producing this intermediate hydroxyethyl TPP. Notice that this part of the molecule is this part. Now, this substance then undergoes reaction with lipoic acid. Okay, with lipoic acid in the process, see, forming this intermediate in which the sulfur-sulfur bond has been cleaved. Okay, by the way, this first step is catalyzed by pyruvate dehydrogenase. Okay, the second step is catalyzed by dihydrolipoyl transacetylase, which is the reaction between hydroxyethyl TPP and oxidized lipoic acid, this one, process producing the acetyl group is covalently linked to a sulfur. That is a high energy bond, by the way, because that is a thioester bond. Right, its cleavage is highly exergonic, so you have SH. The second, or rather the third step, is a reaction with the coenzyme A, or CoAsh. Now, it is coenzyme A is a molecule that has a thiol group in the end. That's why it's an analog of alcohol. Remember, thiols are analogs of alcohol. Instead of having an OH, they have an SH. All right, they have an SH. Okay. Um, um, uh, then what is the next step? The next step, remember, once a coenzyme A reacts with this sulfur, you end up with the process is trans-esterification, because one um, thioester is replaced by another thioester. This is another thioester, another high energy molecule. So in the next step, notice that the S carbon bond is replaced by an SH bond. Effectively reduce the lipoic acid, and this lipoic acid is oxidized back, or a reduced form of lipoic acid is oxidized back into oxidized lipoic acid by reaction with NAD plus. Actually, what happens is that it does not react with NAD plus directly. This is first oxidized by FAD, converting it into FADH2, and then FADH2 is then oxidized by NAD plus to in the process produce NADH and H plus. Now that is accomplished with the enzyme called dihydrolipoyl dehydrogenase. Right? Okay. In summary, two carbon needed at the start of a citric acid cycle is obtained by converting pyruvate to acetyl-CoA. The conversion requires three primary enzymes in the pyruvate dehydrogenase complex. What are these enzymes? The pyruvate dehydrogenase, lipoyl transacetylase and dihydrolipoyl dehydrogenase, right? As well as the cofactors thymine pyrophosphate. We've already encountered thymine pyrophosphate actually when we studied the um, alcoholic alcoholic fermentation. Remember the conversion of pyruvate into acetaldehyde that required thymine pyrophosphate as well. The FAD, NAD plus, and lipoic acid, TPP. These are and even the cofactor coenzyme A, right? These are required for the action of three enzymes: pyruvate dehydrogenase, dihydrolipoyl transacetylase, and dihydrolipoyl dehydrogenase. The overall reaction of the pyruvate dehydrogenase complex is the conversion of pyruvate, NAD plus, and coenzyme A to what? Acetyl-CoA. Acetyl-CoA enters the citric acid cycle. NADH, which is oxidized in the electron transfer chain, and carbon dioxide, corresponding to the first carbon that is removed from from pyruvate. Okay. Okay. Now let's now consider the steps in the citric acid cycle. In step one, there's condensation of acetyl-CoA with oxaloacetate to form citrate. So oxaloacetate condenses or reacts with acetyl-CoA, um, catalyzed with the enzyme citrate synthase to produce citrate and coenzyme A. Now, this reaction, notice, what happens is that the coenzyme A has been removed and that it forms the molecule citrate. Citrate is a meso compound. It is not chiral, right? But in the chiral environment of the enzyme active site, the molecule is discriminated, or the part of the different part. There's different parts of the molecule is chirally discriminated by the active site residues in the chiral active site. The reaction is catalyzed by citrate synthase, an allosteric enzyme that is inhibited by NADH, ATP, and succinyl-CoA. Now, does it make sense given the fact that the citric acid cycle is involved in the production of a lot of ATP? Does it make sense that NADH, ATP, and succinyl-CoA are inhibitors of this enzyme? Yes. Remember, um, a pathway is typically regulated at the beginning, at the end, or at some highly exergonic steps. This is the beginning of the citric acid cycle, so it makes sense that when the molecule, or rather when the system is high in energy, meaning it has a lot of ATP, there's a lot of NADH. NADH, by the way, is oxidized in the electron transfer chain, and the process of oxidative phosphorylation it's used up in the generation of ATP. So when there's already a lot of ATP and when there's already a lot of NADH, the cell is in a charged state. It does not need more ATP or more NADH because it already has a lot of it. Therefore, it makes sense for the citric acid cycle to be inhibited or to be suppressed. Okay, succinyl-CoA is a downstream product of the citric acid cycle, and I've mentioned it a while ago. So when you already have a lot of succinyl-CoA, that means that um, there's a there's a lot of energy, right? There's a lot of succinyl-CoA, and you don't need to produce more of it because um, the citric acid cycle is already operating at its maximal capacity. I mean, it's already term should be that the citric acid cycle is already producing more than the amount of ATP that is needed by the cell. Okay, now oxaloacetate reaction with acetyl-CoA produces a 6-carbon tricarboxylic acid. 1, 2, 3 tricarboxylic acid. That's why it's called tricarboxylic acid cycle. And this is the reason why this is called the citric acid cycle, because you produce citrate in the process. Coenzyme A is released. Again, the first reaction is a highly exergonic reaction. Okay, in step 2, the by the way, this is catalyzed by citrate synthase. In step 2, the citrate is isomerized to isocitrate, and the reaction is catalyzed with an enzyme called aconitase. Now, citrate, as I mentioned, is a chiral, there's no stereocenter, but isocitrate is chiral. There's 2 stereocenters and 4 stereoisomers are possible, but of these, only one of the 4 stereoisomers of isocitrate is formed in the cycle. Again, that is a testament to the stereospecificity of the enzymes. They're capable of forming only one out of the many possible stereoisomers that can be formed from the reaction of a chiral molecule, or from a reaction that produces chiral molecules. Why? Because again, the active site, the nucleophilic residues, and the residues involved in the reaction are arranged in space in such a way so as to favor formation of one stereoisomer and only one stereoisomer over the other. Okay? Now, these are the reactions of the citric acid cycle and the corresponding enzymes that catalyze them. So we're already citrate synthase, so we now consider citrate to isocitrate. We have aconitase. Here is what happens. So citrate undergoes first dehydration. This is catalyzed by aconitase again, so forming a double bond. Here afterwards, it undergoes hydration reaction, where the um, molecule forms isocitrate. The molecule of water, or rather hydroxyl group, is then removed from this carbon, and the molecule of water attacks this carbon atom to, in the process, give you a molecule in which the OH is found now on the carbon atom. Initially, the OH is found on this carbon. Now the OH is found on the other carbon atom. Initially, the hydrogen is found on this carbon. Now the hydrogen is found on the other carbon. So that's just an isomerization reaction, right? So the citrate is isomerized into isocitrate. Okay. Now, in this third step in the citric acid cycle, the isocitrate is oxidized in the process, and then followed by decarboxylation to form alpha ketoglutarate and carbon dioxide. So isocitrate is first oxidized, NAD plus is converted into NADH, and then you form oxalosuccinate. This oxalosuccinate then undergoes decarboxylation to produce carbon dioxide and alpha ketoglutarate. Now, the reaction is catalyzed by isocitrate dehydrogenase. It's an allosteric enzyme which is inhibited by ATP and NADH, and is activated by ADP and NAD+. Now, before we consider the allosteric regulation as such, I want to emphasize that the reaction, unlike the pyruvate dehydrogenase complex, does not require TPP, lipoic acid, and does not require those two. Okay, but like the pyruvate dehydrogenase complex reaction, it involves decarboxylation. Actually, the second molecule of carbon dioxide is lost at this stage, and that NADH is formed. This is actually what the reaction is this now. This is the second reaction where NADH is formed. The first is in the reaction of the pyruvate dehydrogenase complex, right? This is the second reaction where we encounter NADH. By the way, the action of the pyruvate dehydrogenase complex is not part of the citric acid cycle. the formation of acetyl-CoA, in other words, is not part of the citric acid cycle. It is the reaction of acetyl-CoA with oxaloacetate that is the first step in the citric acid cycle. Okay, now ATP and NADH again inhibits this enzyme, the enzyme isocitrate dehydrogenase, because it indicates that there's already a lot of ATP and NADH and that you do not need to produce more of it. But in the state of the cell where it lacks energy and it's desperate for energy, that is, um, when there is an abundance of ADP and NAD plus, then the reaction is enhanced, and that isocitrate dehydrogenase is activated by ADP and NAD plus, which again makes sense because if your system is lacking in energy and it needs energy, then it will have to form more ATP, form more NADH, and it does that by having the ADP and NAD plus activate this step, or the enzyme that catalyzes this step. Okay. Now in step 4, there's again another oxidative decarboxylation. So it's alpha ketoglutarate to succinyl CoA. Now this reaction is similar to what happens in the pyruvate dehydrogenase complex. Remember there, pyruvate is converted into acetyl-CoA. Excuse me. In this case, in step 4, where is it? The let me go back to that because it's not shown here. Here, in this case, the where am I now? Where here? Alpha ketoglutarate. So the alpha ketoglutarate is oxidized to succinyl CoA. As I've mentioned, the reaction or the enzyme that catalyzes this reaction is similar to the pyruvate dehydrogenase complex, have here alpha ketoglutarate dehydrogenase complex, which again requires the presence of gluconic acid, TPP. In this case, NADH is formed, carbon dioxide is formed. That completes the steps that are necessary for the production of carbon dioxide, for the complete oxidation of pyruvate into 3 molecules of carbon dioxide. Remember, the first carbon dioxide is lost here, pyruvate to acetyl-CoA. The second is from isocitrate to alpha ketoglutarate, and then the third is from alpha ketoglutarate to succinyl CoA. Right? NADH is formed as well, so that's the third step. First is here, second is here, third is here, where you form NADH. Alright? Now, let's go back to this. Okay, now, before I proceed, the fluoroacetate is a potent inhibitor of the um, uh, enzyme um, of the enzyme. Let's look at that. The cycle again. Which enzyme does it inhibit? So fluoroacetate is actually an analog of acetate. Fluoroacetate is converted into fluoroacetyl-CoA, so it inhibits citrate synthase, right? Citrate synthase in the process produce fluorocitrate. Now, fluorocitrate inhibits aconitase, and therefore shuts down the process of citric acid cycle. And that is very important because, or that is very important because, where am I? All right, because shutting down a major metabolic pathway that leads to the formation of a lot of ATP will deprive the cell of the energy it needs to perform its essential functions, therefore will cause the organism to die. And that's exactly what happens here. Fluoroacetate is produced by certain poisonous plants, and these plants, um, for example, in the United States, ranchers or those people who, um, what do you call that, who have sheep, all right, they put this poisonous plants outside the just immediately outside the what, what do you call it, the sheep pen, immediately outside the sheep pen, so that when coyotes eat them, the coyotes die, right? Because the coyotes, they tend to what do you call that, to feed or to feast on the sheep. They do not, the ranchers, of course, the sheep owners do not want that. So they put these poisonous plants outside the sheep pen, and so when the coyotes eat them, coyotes drop dead. Okay. Now, the reaction, as I've mentioned, of alpha ketoglutarate um, uh, into what is the product of that? Product is, look at that reaction, into succinyl-CoA is catalyzed with alpha-ketoglutarate dehydrogenase complex, which is, as I mentioned, like pyruvate dehydrogenase, a multi-enzyme complex and requires coenzyme A, thymine pyrophosphate, epoic acid, FAT, and NAD+. Okay, next, the thioester bond of succinyl-CoA is hydrolyzed in the formation of succinate. Right, the two CH2COO groups of succinate are equivalent. Now, this is the first energy yielding step of the cycle in the sense that it produces GTP. Remember, GTP is energetically equivalent to ATP. In fact, it is converted by nucleoside um, uh, diphosphate kinase into GTP. This is the first reaction in glycolysis. I'm sorry, in the citric acid cycle that directly produces ATP, quote unquote. Okay, a substrate level phosphorylation. So succinyl-CoA synthetase catalyzes this reaction. Now it's called a synthetase because the reaction leads to the formation of a new bond, and because it requires high energy phosphate molecule. Actually, the reaction is named for, or the enzyme is named for the reverse reaction, succinate to GTP, because the reaction is reversible. So succinyl-CoA synthetase produces a compound with new bond, that is new bond. The new bond is a carbon-sulfur bond between the coenzyme A and the succinate, and it requires GTP, a high energy compound. Now the overall reaction is just slightly exergonic. Now the next reaction is an oxidation of succinate into fumarate. In that case, a double bond is formed, right? Succinate and the fumarate reaction causes the conversion of NAD to FADH2. Again, this reaction occurs on the inner mitochondrial membrane because the enzyme succinate dehydrogenase is an integral membrane protein. Right? FADH2 is then oxidized by um, oxidized by NAD plus. Okay, then fumarase converts fumarate, or it causes the hydration of fumarate into malate. Malate is oxidized um, by malate dehydrogenase into oxaloacetate. Alright, as you can see here, that is the last step in a citric acid cycle. It regenerates the oxaloacetate. Okay, these are just the same reaction. Succinate converted into fumarate, and then fumarate here, right, is hydrolyzed into malate, and the malate is oxidized into oxaloacetate. Reaction is ready, or the cycle is ready for another round of reaction. Okay. Now, the oxidation of pyruvate forms carbon dioxide and ATP. It is very important. At this point, you have to realize that the molecules of carbon dioxide, or rather of glucose, in this case, have been completely oxidized into carbon dioxide because the three molecules of pyruvate have been converted already into carbon dioxide. Now, in the case of the pyruvate dehydrogenase complex, all right, this is the reaction. Citric acid cycle, this is the reaction. So when you add them up, you end up with pyruvate plus 4 NAD plus 1 FAD plus 1 GDP plus 1 inorganic phosphate plus 2 molecules of water produces 3 carbon dioxide, 4 NADH, 1 FADH2, 1 GTP. Now, 1 GTP is equivalent to 1 ATP. 4 NADH is equivalent to 10 molecules of ATP because one molecule of NADH is oxidized in the electron transfer chain and is responsible for the production of um, 2.5 moles of ATP, while 1 mole of FADH is responsible for the production of 1.5 moles of ATP. Since we have 4 times 2.5, that is 10, 1 FAD is 1.5, so we have 1 ATP here. Total is 12 ATP per, rather 12.5 ATP per pyruvate. But remember, we have 2 molecules of pyruvate from 1 molecule of glucose, so that amounts to how much? To 25 ATP. Alright, that is remarkable. Now we have 25 ATP just from pyruvate into carbon dioxide, compared to what? Just two molecules of ATP from glycolysis. Remember, this is not yet complete because if you have oxidative respiration, then that includes glycolysis and the citric acid cycle. So we have 2 molecules of ATP from glucose to 2 molecules of pyruvate. So that's already 2. But we already produced there 2 NADH, right? 2 NADH molecules. So that's 2 times 2.5 is 5 plus 2, 7 plus 25. So it's 32 ATP. Now, depending on the cell, that's either 30 or 32 because the NADH that is produced from glycolysis in the cytosol is transported into the mitochondria via different shuttle systems, depending on whether you have liver or a muscle cell. So it can be 30 or 32 ATP. In that case, NADH is equivalent. We'll learn later why that is the case. But what I'm trying to emphasize here is the fact that now you're forming a lot of ATP, 30, 32 compared to the anaerobic glycolysis, where molecular glucose is converted into lactate, where you produce just 2 molecules of ATP for every molecule of glucose, right? This is remarkable. Okay, now in summary, in the citric acid cycle, in the pyruvate dehydrogenase reaction, 1 molecule of pyruvate is oxidized to 3 molecules of carbon dioxide as a result of oxidative decarboxylation. The oxidations are accompanied by reductions involving NAD plus to NADH, FAD to FADH2, and GDP is phosphorylated to GTP. Okay, now let us now consider, or let us now study the control points, the regulation of the citric acid cycle. As I've mentioned, there are 3 points of control within the cycle. The reaction of the citrate synthase, it's here. Remember this reaction, citrate synthase catalyzes acetyl CoA, oxaloacetate reaction to produce citrate. The reaction isocitrate catalyzed by isocitrate dehydrogenase that converts isocitrate to alpha ketoglutarate. Remember, and alpha ketoglutarate dehydrogenase complex. Okay, now let us look at this cycle, because here we emphasize the control points. Okay, the one with this red asterisk, quote unquote, are the steps that require electron acceptors, either NAD plus or FAD. Pyruvate to acetyl CoA. This is strictly not part of the citric acid cycle, right? The reaction catalyzed by pyruvate dehydrogenase complex. Because the citric acid cycle is acetyl CoA to oxaloacetate, this one. But the pyruvate dehydrogenase complex reaction is, of course, the reaction that funnels acetyl CoA into the citric acid cycle, so they're intimately related. The This um, symbol is for inhibition of reaction, and the X marks the control points for the reaction. Okay. All right. Okay. So let us consider this. Okay. I think I've already mentioned some of this during the discussion, but just so to emphasize, um, uh, hmm, the reaction catalyzed by citrate synthase, acetyl-CoA oxaloacetate producing citrate, is inhibited by ATP and NADH. You've already mentioned that before, right? When there is already a lot of ATP and NADH, and the cell is said to be in a charged state. It is in a state where it already has a lot of energy sources, and therefore a lot of ATP, a lot of energy, then therefore it does not need to produce more ATP and more NADH. So ATP itself and NADH inhibits this reaction. Succinyl-CoA, a downstream product, also inhibits the reaction with citrate synthase, signaling that hey, you have to stop because we already have a lot of downstream products here, succinyl-CoA, and so the citrate synthase is said to slow down, still to slow down. Citrate, the very product of a reaction, inhibits citrate synthase, again suggesting that there's a lot of citrate and reaction will have to slow down. Now, isocitrate dehydrogenase is inhibited by ATP and NADH. Again, that makes sense because a charge state, the cell in a charged state does not need to produce more ATP and NADH, but it's stimulated by ADP and NAD+. Why? Again, because in that case, when you have low ATP, we have a lot of ADP and NAD+, and that's the cell in its um, energy deficient state. So you have to produce more ATP and more NADH. So the ADP and NAD+, which are abundant in the low energy state, activates isocitrate in order for the system to produce more ATP and more NADH process, giving the cell more energy. The third is the reaction alpha-ketoglutarate to succinyl-CoA. It is inhibited by ATP and NADH and succinyl-CoA. Again, that makes sense. We already have a lot of ATP and NADH. You do not need to produce more ATP and NADH. Succinyl-CoA, it's a product of a reaction. When you have a lot of succinyl-CoA, it means that the citric acid cycle is operating at its maximal efficiency, and that it does not need to produce more of succinyl-CoA because there's already a lot of it. Okay. Um, Here is shown the energetics of the citric acid cycle. And you're going to notice that overall, as you have expected, the citric acid cycle is highly exergonic. When you add all the reactions up, you're going to end up with negative 7.7 kilojoules per mole. Actually, it's highly exergonic, right? Um, okay, I've been mentioning something about regulation already, mentioning um, charge state and so on, and energy deficient state. Here is a relationship between the metabolic state of a cell and the ATP to ADP and NADH to NAD plus ratio. So cells in a resting metabolic state need and use comparatively little energy. You don't need it because they already have high ATP, and low ATP levels imply high ATP to OE to ATP ratio, right? High NADH, low NAD plus levels imply high NADH, excuse me, over NAD plus ratio. This is a cell in a resting metabolic state, or a cell is at ease. I already have a lot of ATP, I already have a lot of NADH. All right, so it does not have any more need for energy because it has a lot of them, and secondly, because it's in a resting metabolic state. But if a cell is in an active metabolic state, it uses up a lot of its ATP and a lot of its NADH, and so the system or the cell must be able to produce more ATP and more NADP, more NADH in that case. So need and use more energy than resting cells. They have low ATP to ATP ratio. They have low NADH to NAD plus ratio. And as such, they will need to produce more ATP and NADH. There is actually a very interesting cycle, the glyoxylate cycle, that bypasses the oxidative steps in the citric acid cycle. It bypasses those two steps. And the glyoxylate cycle is found in plants and some bacteria. Um, there may be a modification of the citric acid cycle to produce four carbon dicarboxylic acids and eventually glucose. Um, this is the reason why plants and certain bacteria are able to convert fats into carbohydrates. Unfortunately, this cycle does not happen in mammals and in animals. That's why we, to the chagrin of many, cannot convert carbohydrates, or rather, cannot convert fats into carbohydrates. We're able to convert carbohydrates into fats, but not fats to carbohydrates. The glyoxylate cycle bypasses the two oxidative decarboxylations of the citric acid cycle. Instead, it routes isocitrate via glyoxylate to malate. Alright, the key enzymes in this cycle are isocitrate lyase and malate synthase. Hopefully very soon. Okay, here it is. So the isocitrate, remember here. So this is the citric acid cycle. So what happened is that the isocitrate here, instead of forming succinate, in a reaction catalyzed by, there are many reactions here, right? Isocitrate, um, isocitrate dehydrogenase, and you have aconitase converts isocitrate to succinate, and alpha ketoglutarate dehydrogenase. There are a number of steps there. Um, in the case of, let's look at this. In the case of the glyoxylate cycle, the isocitrate is converted directly to succinate and glyoxylate by an isocitrate lyase. Okay, isocitrate is converted into glyoxylate and succinate by isocitrate lyase, in the process bypassing the two steps conversion of isocitrate to alpha ketoglutarate and then alpha ketoglutarate to succinyl CoA to succinate. The two decarboxylation steps have been bypassed. Then afterwards, the glyoxylate reacts with acetyl CoA to produce malate, catalyzed by malate synthase, and then malate is oxidized to oxaloacetate. As you can see here, glyoxylate acetyl CoA producing malate. Now, we've been saying that the importance of this stems from the fact that, if you remember, what is the product again of the glyoxylate cycle? Isocitrate, glyoxylate, then glyoxylate reacts with acetyl CoA to produce malate, which is oxidized to oxaloacetate. Oxaloacetate, if you remember, is involved in the gluconeogenesis, the production of glucose, right? Then you're going to say, hmm, How exactly does or does this cycle allow for the conversion of fats into carbohydrates? Okay. Um, let's take for example a germinating seed. The germinating seed needs, as it germinates and as it becomes a seedling, it it requires a lot of energy, right? And not only that, it also requires building blocks because plants are made up of carbohydrates like cellulose. All right. Okay. So what happens is that these seedlings have a lot of fat stores. So as they germinate, the fats are oxidized to acetyl CoA, which we're going to learn the next couple of discussion. They are oxidized into acetyl CoA. That acetyl CoA then during remember they have glyoxylate cycle. Reacts with the glyoxylate to produce malic and then oxaloacetate. That oxaloacetate is funneled through gluconeogenesis into glucose. Okay. Now, the germinating seed also needs energy, and it it's able to synthesize so a lot of ATP through the citric acid cycle because although there's a glyoxylate cycle here, this seedling can also accomplish the citric acid cycle. Do not think that glyoxylate cycle, when it occurs, the citric acid cycle does not occur. Metabolism is a complex, interconnected process. So glyoxylate cycle occurs there, citric acid cycle occurs there as well. The germinating seed needs a lot of ATP, so citric acid cycle provides ATP for it. Now, the question is, what about humans or what about mammals or animals? We cannot do that because if we remember we do not have glyoxylate cycle. If we convert all of our or a significant fraction of our oxaloacetate into glucose, the oxaloacetate, right, will be depleted, and so the citric acid cycle will be compromised. We cannot we cannot compromise it because we need ATP, and also we cannot convert fats. That's that's the reason why we cannot convert fats into carbohydrates because we do not have the glyoxylate cycle. The glyoxylate cycle provides you with an extra oxaloacetate that you can use for the synthesis of glucose. Since we do not have glyoxylate cycle, we cannot provide that extra oxaloacetate. Okay. Unfortunately, that's why um, fats cannot be converted into carbohydrates in humans. Carbohydrates can be converted into fats. Okay. The glyoxylate cycle takes place in plants in um, what we call glyoxysomes, specialized organelles devoted to this cycle. In yeast and algae, it occurs in the cytoplasm. Okay, the glyoxylate cycle helps plant grow in the dark. Seeds, as mentioned, are rich in lipids, which contain fatty acids. During germination, plants use the acetyl-CoA produced in fatty acid oxidation to produce oxaloacetate and other intermediates for carbohydrate synthesis. Once plants begin photosynthesis and can fix carbon dioxide, glyoxysomes disappear. Right? Why is that the case? Because plants are able to generate their own glucose now using photosynthesis. So glyoxylate cycle is no longer needed, so the glyoxysomes disappear. Okay, as I mentioned before, the citric acid cycle is a catabolic cycle. It's also an anabolic cycle. In fact, it's what we call an amphibolic cycle. So the catabolism of proteins give rise to alpha keto acids, the carbon framework, and these alpha keto acids can enter the citric acid cycle. Catabolism of carbohydrates, as I've already mentioned, we've already learned. glucose into pyruvate to acetyl-CoA. Fatty acids, the oxidation of fatty acids, the beta oxidation of fatty acids, which I'm sure many of you might be interested. Um, Beta oxidation of fatty acids produces acetyl-CoA. They all feed into the citric acid cycle at one or more points. Let's just consider the lipids. Lipids, the process of beta oxidation, produce acetyl-CoA. Carbohydrates, remember, glycolysis produces phosphoenolpyruvate, and then pyruvate. This pyruvate is also ultimately converted into acetyl-CoA. Now, what about the amino acids? Amino acids like threonine, alanine, serine, cysteine, and glycine, their carbon frameworks are ultimately converted into pyruvate. Asparagine, aspartate, um, uh, they are converted into what? Oxaloacetate and malate. Remember, malate is part of the citric acid cycle. Tyrosine and phenylalanine are oxidized or metabolized into fumarate, which then enters the citric acid cycle. Threonine, valine, isoleucine, and methionine converted into succinyl-CoA, which is part of the citric acid cycle. Proline, arginine, glutamine, and histidine converted into glutamate, which is converted into alpha-ketoglutarate, which is again part of the citric acid cycle. This is what's happening. Carbohydrates, lipids, and amino acids are metabolized and used as a source of energy through the citric acid cycle. Remember, a person who is fasting becomes very, very thin. At first, the first stages of the fast, the um, glycogen is being consumed. At the latter stages, remember, the liver maintains blood glucose level. The blood glucose level drops, and in order to prevent that from happening, glucose has to be produced in the process of gluconeogenesis in the liver, right? And there are a number of sources for that, like the proteins, the amino acids of the proteins, they can be used for gluconeogenesis, right? In the end, um, uh, even the proteins themselves, the amino acids of the proteins are used up and used as a source of energy in the citric acid cycle. That's why if you're going to notice people who are fasting become abnormally thin. They're very, very thin. Okay. Um, so in summary, all metabolic pathways are related and all of them operate simultaneously. We're studying them one at a time. It's only as a matter of convenience. It does not mean that these processes do not occur all at the same time. They occur all at the same time. And the cell adjusts and determines which of these reactions have to be, or cycles or pathways have to be activated and have to be deactivated depending on its need. In catabolic pathways, nutrients, many of which are macromolecules, are broken down to smaller molecules such as sugars, fatty acids, and amino acids. Small molecules are processed further, and the end products of catabolism frequently enter the citric acid cycle, which plays a key role in metabolism. That's why when we said that when you plot the entire metabolic pathways in the living system, the citric acid cycle will be more or less at the center or at the heart of it. Okay, the citric acid cycle is also an anabolic pathway, therefore it's important in anabolism. So the citric acid cycle is a source of starting materials for the biosynthesis of other compounds. If a component of a citric acid cycle is taken out for biosynthesis, it must be replaced. For example, oxaloacetate, for example, is replaced by the carboxylation of pyruvate. Remember, the synthesis of gluconeogenesis, rather in the process of gluconeogenesis. Remember, pyruvate there is converted into glucose. Oxaloacetate in the process is used in the formation of phosphoenolpyruvate. That means that the oxaloacid, the phosphoenolpyruvate then is funneled to the production of glucose in the process of gluconeogenesis. Now, The oxaloacetate has to be replenished, and the reaction that replenishes that, like the carboxylation of pyruvate, um, are called is called anaplerotic reaction. A reaction that replenishes a citric acid cycle intermediate is called an anaplerotic reaction. Now, why do you need to replenish the citric acid intermediates? You need to replenish that because you need the citric acid cycle going, right? You cannot deplete the intermediates of the citric acid cycle, otherwise it will shut down and it will cause you production of ATP, and it's going to lead to death if the citric acid cycle is shut down. As I've mentioned, a number of inhibitors, um, the inhibitors that inhibit the citric acid cycle lead to death because that causes you to produce no more ATP and therefore no more ATP to power necessary life processes. Okay, um, uh, these are this is citric acid cycle, as I mentioned. Um, uh, the anaplerotic reactions are as follows. So, for example, I'm sorry, the anabolic reactions. For example, alpha ketoglutarate is transaminated to glutamate. So, the glutamate, that's an example of an anabolic reaction because glutamate is an amino acid that is component of proteins. Now, what else? Um, Oxaloacetate, this can be used for the synthesis of glucose, right? So that's an anabolic reaction. What else? Acetyl CoA can can be converted into oxaloacetate via the glyoxylate cycle. So green plants and some bacteria can do this. Acetyl CoA to pyruvate. Um, Mammals cannot do this again because they do not have the glyoxylate cycle. Okay, all right. Mhm. Remember, the glyoxylate cycle converts isocitrate into glyoxylate and succinate. Glyoxylate then reacts with acetyl CoA to, in the process, produce oxaloacetate. And oxaloacetate um, is used up in the synthesis of glucose, right? Okay. All right. All right. So here, as I mentioned, this is the citric acid cycle, and this emphasizes the anabolic pathways where citric acid cycle is involved. So malate, for example, malate is funneled for the synthesis of carbohydrates. Because remember, the malate can be oxidized to oxaloacetate, then phosphoenolpyruvate, and so on and so forth. Now, what else? Hmm, okay. How do you convert? How do you produce malate? Again, remember, pyruvate goes into the mitochondrion, which is carboxylated into oxaloacetate. Oxaloacetate can either be converted into phosphoenolpyruvate. If it does, then the phosphoenolpyruvate is transported into the cytosol for gluconeogenesis. But gluconeogenesis requires NADH. That is why the other fate of oxaloacetate is it's reduced by mitochondrial NADH, and once it is converted to malate, that malate then is transported into the cytosol. The malate then undergoes oxidation, which reduces NAD plus to become the NAD plus becomes NADH, and so the NADH is used in the synthesis of glucose. Okay. Now, what about lipid anabolism, the formation of lipids? Lipid anabolism begins with acetyl-CoA and takes place in the cytosol. All right. So the Acetyl-CoA molecules, remember, acetyl-CoA is an intermediate in the citric acid cycle. Acetyl-CoA reacts with citric acid, right? So acetyl-CoA is produced mainly in the mitochondria from catabolism of fatty acids and carbohydrates, right? If, say, for example, you're eating a lot of carbohydrates, like you're eating a lot of rice or eating a lot of bread, and that means you're forming a lot of pyruvate, and the pyruvate is converted into a lot of acetyl-CoA. Now, when you have a flood of acetyl-CoA, because if you're eating a lot of carbohydrates, that acetyl-CoA are going to be transported into the um, uh, lipids or fatty acids, into fats, in other words. That's why people get fat when they eat too much. So the what happens is that acetyl-CoA um, is essentially um, transported. Essentially, it is transported. Um, I have to I have to clarify that because what happens is that the um, uh, anabolism acetyl-CoA into lipids is happening in the cytosol, but acetyl-CoA is formed in the mitochondrions, and there has to be a way by which you'll be able to transport acetyl-CoA into the cytosol, right? And that acetyl-CoA, once it's transported into the cytosol, is used up in the synthesis of fats or fatty acids, which become fats in the end. Right, an indirect transfer mechanism exists involving citrates, so the citrate reacts with coenzyme A and ATP to produce acetyl-CoA, oxaloacetate, ADP, and inorganic phosphate. All right. Okay. So where does this citrate come from? This citrate comes from the oxaloacetate and acetyl-CoA reaction in the mitochondrion, and the citrate is transferred into the cytosol, which decomposes into acetyl-CoA and oxaloacetate. Now, acetyl-CoA is then used up in the synthesis of lipids, of fatty acids. The oxaloacetate fast form provides a means for the production of the NADPH needed for biosynthesis. Um, how? Because the oxaloacetate, remember, is reduced into malate when the malate reduces NADP plus to produce pyruvate, carbon dioxide, and NADPH. So the net effect of these two reactions is a replacement of NADH by NADPH, and it is important because NADPH is the one that is needed for the synthesis of fatty acids. While there are some NADPH produced by this means, its principal source again is still the pentose phosphate pathway. So that's why we mentioned before in our study of the pentose phosphate pathway, but it is uh, primarily the pathway that produces NADPH, and that it is the pentose phosphate pathway is an anabolic pathway in the sense it provides um, reducing equivalents for anabolism. The anabolic reactions that produce amino acids and many other biomolecules begin with citric acid cycle molecules that are transported into the cytosol. Okay, right. So there's a summary of the anabolism in the citric acid cycle. Okay, you can just look at that. Phosphoenolpyruvate is transported into um, uh, the oxaloacetate is converted into phosphoenolpyruvate. Phosphoenolpyruvate transported into the cytosol to produce carbohydrates. What else? Malate is used for the synthesis of oxaloacetate, which by transamination reaction produces aspartate. Aspartate is produced in the synthesis of pyrimidines, lysine, threonine, methionine, isoleucine, and asparagine. In other words, amino acids and the pyrimidine bases. What else? Um, um, Succinyl-CoA used in the synthesis of alanine. Alanine becomes used in the synthesis of porphyrins, the heme of hemoglobin, for example. Alpha-ketoglutarate by transamination produces glutamate, and glutamate is used in the synthesis. It's already an amino acid. It can be used in the synthesis of proline, arginine, and glutamine. Isocitrate produces alpha ketoglutarate, which again transamination glutamate, citrate, as I mentioned, citrate is transported into the cytosol, producing acetyl-CoA, that's used in the synthesis of fatty acids and steroids, or leucine and lysine. Pyruvate itself, right? And then how do you obtain pyruvate, or where do you obtain pyruvate? From oxaloacetate, remember. Oxaloacetate, pyruvate. Pyruvate is used in the synthesis of amino acids like lysine, valine, isoleucine, alanine, glycine, serine, and cysteine. Cysteine. All right. Okay. All right. Okay. Now, in summary, the citric acid cycle plays a central role in anabolic pathways as well as in catabolism. Pathways that give rise to sugars, fatty acids, and amino acids all originate with components of the citric acid cycle. Okay? Alright. Now, as I mentioned before, the process of the citric acid cycle is intimately associated with the electron transfer chain and the process of oxidative phosphorylation. Citric acid cycle is part of the aerobic metabolic processes because of its link to the electron transfer chain and oxidative phosphorylation. And what are its links? What links the citric acid cycle exactly with electron transfer chain and oxidative phosphorylation? The NADH and the FADH2, the reducing equivalents that are formed in the citric acid cycles, ultimately pass their electrons to oxygen, the ultimate electron acceptor in the reactions of the electron transfer chain, ultimately leading to the formation of ATP for the process of oxidative phosphorylation. Right? Alright, so that finishes our discussion of the citric acid cycle. In the next meeting, we're going to discuss the electron transfer chain, oxidative phosphorylation, as well as the process of photosynthesis and the lipid beta oxidation of lipids and the synthesis of lipids. Right? Now, I hope that you have learned much in today's discussion.